Hi everyone, I'm Kenzie Cromer, and my literature review is about prenatal androgen exposure in lemurs, how it relates to female social dominance. So, most mammals exhibit male social dominance. In contrast, female social dominance is found in almost all living members of Lemuridae and is characterized by male-like behaviors and morphology. However, the hormonal mechanism for female social dominance is contested, as it's not clear which hormones are responsible. So, our hypothesis is that elevated prenatal androgen exposure in females leads to organizational masculinization of the brain, setting the stage for female social dominance later in life. Now, for our methods, I'm going to zoom in a little. So the methods for these three studies were very similar. Drea and Grieve et al. Used ring-tailed lemurs, or Lemuricata, in their studies, and Petty and Drea used two subspecies of brown lemurs in their study. One subspecies displays female social dominance, and another one which displays codominance between the sexes. Blood samples from the subjects were obtained on a bi-monthly schedule, and then serum hormone levels of A4, testosterone, and estradiol were measured using commercial radioimmunoassay kits. In addition, Petty and Drea and Grieve et al. Looked at behavioral measures of dominance. Social behaviors were measured using continuous focal sampling and included dominance interactions such as cuffing, lunging, chasing, and biting. Moving on to our results, going to start at the left. So to the far left, we have figure 1, which is from Drea's study on pregnant Lemuricata. Just to orient yourself, there are two hormone concentration levels being measured here in nanograms per milliliter, testosterone and A4. There are three categories being compared. First, we have non-pregnant females, which are represented by the teal bars. We have pregnant females, which are represented by the white bars, and we have adult males, which are represented by the black bars. As you can see, adult male lemurs have significantly higher levels of testosterone, but pregnant females have significantly elevated levels of A4 relative to non-pregnant females. Now, in the middle panel, we have another bar graph. We are again looking at two hormones, A4 on the left and testosterone on the right. The categories being compared are females from the two subspecies of brown lemur discussed earlier. Dominant eulemurs females are represented by the light teal bars, and codominant eulemur females are represented by the dark teal bars. Here you can see that codominant females have significantly lower levels of A4 and testosterone compared to dominant eulemur females. On the far right, we have a graph depicting A4 levels in male and female Lemuricata, again in nanograms per milliliter, from 3 to 30 months of age. On the left, we have females, and on the right, we have males. Each gray line represents an individual, and the yellow lines represent the mean A4 level for each sex. And you can see that A4 levels start to peak in females around 10 months of age, while in males there is a gradual but steady rise in A4. One important thing to note is that different y-axis scales are used for each panel to highlight variation within the sexes. So the males do have elevated levels of A4 throughout this 30-month time span. From these results, it can be concluded that A4 plays an important role in the masculinization of female lemurs and likely exerts most of its organizational effects during early development, which supports our prenatal androgen exposure hypothesis for female social dominance. Heightened androgen levels are not necessary for male-like behavior to occur in adult females. which also points to a strong organizational effect of androgens during gestation. Now, estrogen and testosterone likely do help maintain an aggressive phenotype during adulthood, but A4 sets the blueprint early in life. For limitations, the most obvious one are these studies' small sample sizes. As you can see here, the smallest sample size is 11 individuals, the largest is 28. So all lemur species are endangered. For this reason, a large sample size is rarely possible. Another limitation is that all the studies relied on androgen concentration rather than receptor density and sensitivity. It's possible that masculinization and female social dominance are mediated by sex differences in receptor distribution rather than absolute hormone levels, which is what was being measured. So the take-home message here is that elevated A4 levels do provide evidence for the hormonal mechanisms of female social dominance, but masculinization seems to occur during critical periods of fetal development. Now, with this in mind, my proposed study is to figure out whether A4 is acting directly on the brain or indirectly. And here is a figure showing a proposed mechanism for how A4 might work directly with receptors. Now, it's important to note that A4 is converted to testosterone through the enzyme 17-beta hydroxysteroid dehydrogenase. I hypothesize that female Lemuricata have greater activity of this enzyme in brain tissue, and we could potentially measure this in individuals who have died of natural causes.